Welcome to Nanolens. Have you ever wondered how scientists look deep inside polymer and biomaterials at the nanoscale? The secret isn't a better microscope, it's a slice 1,000 times thinner than a human hair. In this video at Nanolens, we're breaking down the Ultramicrotome, what it does, why it's essential for TEM, and how it performs the impossible task of slicing the unsliceable. Stick around till the end to see the magic happen. Ultramicrotomy is a critical sample preparation technique used in materials science and biology to slice samples into ultra-thin sections, typically nanometer scale thickness, for analysis under a transmission electron microscope, TEM. This is basically a meat slicer, but instead of meat, it cuts biological and polymeric samples. Why do we need to slice these samples? Because electron beams cannot penetrate thick samples, ultramicrotomy is essential to achieve the required transparency. The device consists of two main parts, the sample holder and the knife, both enclosed in a protective housing. Right next to the knife is a water bath where the slice sections are collected. Let's see how this machine works. First, the device needs to be turned on. The operator powers on the control unit and activates the illumination system via the touchscreen interface. Proper lighting and stable electrical controls are critical because precision alignment requires clear visibility under the built-in stereo microscope to prevent damaging expensive diamond knives. The sample holder base and the sample holder are mounted, and the sample is securely locked into the specimen clamp. The stereo microscope is then adjusted for optimal viewing. Securing the sample tightly is vital. Any mechanical drift, vibration, or loose clamping at this stage will cause chatter marks or uneven thickness in your final sections, ruining the TEM grid preparation. The surface of the resin block containing the embedded sample is trimmed to expose a flat, uniform surface. Facing off removes excess resin to get close to the region of interest. Achieving a smooth, flat face ensures that subsequent ultra-thin cuts will maintain uniform thickness across the entire section. Vertical slicing cycles begin, and the operator shapes the block into a specific trapezoidal geometry. Trimming the block into a trapezoid is a standard best practice in ultramicrotomy. The wider base facing the bottom helps stabilize the ribbon of sections as they float onto the water bath of the knife boat, while the top edge narrows down to the precise region you want to image. The sample holder is loaded into the mechanical arm, and the knife holder is positioned on the stage. This final stage integrates all components: the diamond knife, filled with distilled water to catch the floating sections, and the trimmed sample block, preparing the system for precise, automated advance feeding and high-resolution sectioning. Now the knife is gently pushed toward the instrument by hand until it reaches the desired position. Sample and knife positions are inspected, angles are adjusted, and the system is prepared for precise sectioning at room temperature. Precise adjustment of the knife angle and clearance distance is critical. Even a minor alignment error can cause chatter marks on the sample surface and ruin the sections. Next, the knife boat is filled with distilled water. The distilled water acts as a flotation medium for the ultra-thin sections. Following sectioning, the sections remain floating on the water surface, where they can be easily collected by the operator using a TEM grid. The sample is controlledly advanced toward the knife at preset thicknesses to produce regular slices. The sample is automatically advanced by the instrument with extreme precision to carve out layers possessing a consistent and uniform thickness. The sectioning process is then started. As the specimen mounted on the specimen arm passes across the knife edge, ultra-thin sections are produced. These nanometer-thick sections float on the surface of the water in the knife boat, ready for collection. The sectioning speed and other important operating parameters can be adjusted and modified during the sectioning process. The up and down movement of the specimen arm is what performs the actual sectioning of the specimen. The delicate slices are typically floated onto a liquid bath, like water, allowing them to be safely retrieved and mounted onto specialized microscope grids without structural damage. Although the sections are very small, they can still be observed with the naked eye. The section thickness can be controlled by reducing the sectioning speed and adjusting other operating parameters. Variations in section thickness are indicated by interference colors. Experienced operators can use these colors to make reliable estimates of both the thickness and the quality of the ultra-thin sections. The generated sections are transferred and collected for subsequent laboratory imaging and analysis steps. For more content on topics like these, follow the Nanolens channel